Good morning. My name is Brian Haynes. I work for Fluxion Biosciences, supporting the BioFlux product. And today it's my pleasure to introduce Dr. Jonathan Finkel. This story actually spans close to a decade and goes around the world. Dr. Finkel started his bachelor's degree at St. Olaf College, studying chemistry in Norwegian, then finished a PhD at the University of Madison, Wisconsin. While he was a postdoc at Carnegie Mellon University, he was one of the first BioFlux customers and was actually the inspiration behind using a torque screwdriver for the BioFlux 200 system. There he studied fungal pathogens and then later became faculty at the Carnegie Mellon University in Qatar. He's the reason that we had the first BioFlux system set up in the Middle East. And then later took a position at the famous ATCC, or American Tissue Type Culture Collection, and is now an assistant professor at the University of Detroit Mercy. I want to stress today the importance of this lecture from a public health perspective. In terms of fungal infections, both my grandparents had what they call thrush in their mouth in response to chemotherapy when they were receiving, end of life or extending life chemotherapy towards late stage cancer. People are remarkably susceptible to fungal infections that have issues like living with HIV or AIDS, organ transplants, cancer patients, hospitalized patients, stem cell transplant patients, or patients taking medications that affect the immune system. There are over 1 billion cases of fungal infections every year, and the total cost, at least in the United States, is over $5 billion per year. And with that, it's my pleasure to turn this over to Dr. Finkel.
Good morning, everybody. Thank you for joining me today, and thank you, Brian, for that wonderful introduction. I'm excited to be able to share with you guys how I've been able to use the Fluxion BioFlux 200 to study adherence and biofilm formation, not only in Candida albicans, as the title would suggest, but in many other fungal species. Again, Jonathan Finkel at the University of Detroit Mercy. So since I was looking at our list of attendees, and it was quite varied in different fields, and so I figured I would just give a little bit of background about what a biofilm is and why is it important to study. Well, if we take the Wikipedia or dictionary definition of a biofilm, you find that it is a surface-associated microbial communities that are surrounded by an extracellular matrix. Now, like with most definitions, that can be a little dense. So to summarize, if we think about, we have a surface, and that surface can attract a microbial entity, and that microbe will divide and divide and divide. It then encases itself in an extracellular matrix, which is depending on the species, a beta-glucan, sugars, polysaccharides, even some DNA actually in there, and that actually helps to protect the biofilm from the environment and also from competing microbes, or in the case of a pathogen, from the human host. Now in nature, most microbial communities are actually in a biofilm. So why is a biofilm something we should care about? Well, the reason we see biofilms on devices is that if we think about tissue, when a microbe comes and it binds to a tissue, the tissue has evolved different defense techniques. And basically this, for instance, induces cytokines. And the cytokines then recruit defensive cells, which come and destroy our poor microbe. Well, actually a good thing for us, but for the microbe, not so much. Now, if we think about an abiotic surface, this surface has no cytokines. And because there's no cytokines, there's no defense mechanism that can be recruited. And this allows for the microbe to replicate, or to divide, and over time form a biofilm on that device. Now, my research focus began with and is still pursuing understanding the pathogen Candida albicans. Now, Candida albicans is commensal and pathogenic, and we find it in two major forms. In one form, we have the yeast cell, which I hope everyone can see my pointer here. You have a mother cell, and then you have the daughter cell, which buds off of it. And the yeast will bud and bud and bud and bud and bud. Now, we also find a filamentous form, or what we call a hyphal form, which will look like this. You have your mother cell, and you have your long protruding cell. Now, this hyphal is the predominant form when we look at when it becomes pathogenic within the body, systematic. Basically using this to wiggle its way in between cells or punch through cells to further the infection. Now, it's an opportunistic pathogen, which is a very important thing to understand. Of those of us here today, 90% of us will have Candida albicans somewhere in our body, and most likely in our gut. And that 90% can change daily. So some of us, they'll have it today, may not tomorrow, and those that didn't will have it today. It's not a problem in normal, healthy adults, but there are certain risk factors that can result in it becoming pathogenic. Now, Brian did kind of hit some of the immunocompromised status aspect of this, and I think it's important to stress, though, that I think most individuals, when you say immunocompromised status, we unfortunately think of, we think of the unfortunate status of HIV causing AIDS. And it is a primary source of immunocompromise. But there's many other things that can result in an immunocompromised status. And that includes cancer treatments, like Brian was stating, but it's also diabetes can result in it. Pregnancy. If you have intensive surgery can result in some immunocompromised status. And then, of course, transplants and other treatments that result in reduced immune system. Now, another risk factor is antibiotic therapy. So as I mentioned, we would find Candida albicans in most of our guts. And with heavy antibiotic therapy, basically what happens is that you then remove all the bacteria, or most of the bacteria in the gut, and you've basically left a reservoir or an empty space for the fungi to take over. And that's what they can do. And then they become pathogenic and cause all kinds of problems. Now, there's implanted devices. Implanted devices, I have to say, is probably one of the greater techniques that have been developed for the betterment of health, but they also run the risk of causing trouble. Implanted devices, of course, would include your pacemakers, your stents, your artificial valves. My mother has a new knee. My mother-in-law has a new hip. Yes, and so all these devices can make you more susceptible to an infection. Now, unfortunately, there are few classes of antifungal drugs that are available for treatment. And even with treatment, we see a 30% mortality rate for invasive infections of Candida albicans. Now, the reason that biofilms are important is because biofilm formation is the major cause of device-associated infections. So you'll have an implanted device. That device will have a biofilm, and it then becomes very difficult to treat and can lead to invasive infections or systematic infections. Now, a lot of work has been done in vitro, and a lot of work has been done in vivo. But initially, we started out with in vitro work, and there's been a lot of great work that was done before I even started college on how does a biofilm form in vitro. And the basic idea is that we would have the first step of adherence, where yeast-formed cells would adhere to a substrate, as we see here. Now, as these cells bind, this is, again, an abiotic surface, so there's no cytokine response, no defense, and the cells can keep dividing. Now, as they divide, they hit the initiation step where they start to form filamentous cells, such as the hyphal cell that I showed in the previous slide. As the biofilm matures, we find that the cells, we have a large amount of yeast-formed cells at the bottom. The biofilm itself is interspersed with hyphal cells with some yeast cells on the top, and the whole thing is encased in an extracellular matrix. Now, this is not the end of our story of our biofilm, because this in itself would not be that detrimental to human health. It's this last step where the biofilm itself will release yeast cells to further colonize the host. So this could basically be moving along to a different part of, let's say, in the medical sense, the device in which it formed on. So now you're forming on a new part of the device, or this could be basically being released into the body to cause potential systematic infections. Now, one of the things that we've noticed is that as you go from left to right here, you see increased drug resistance. And part of the major problem with this is that you can treat the cells that are being dispersed and causing the systematic infection, but you're not able to eliminate or eradicate that biofilm that is already formed. And since that biofilm is there, after treatment is concluded, you basically end up with an infection occurring again. Now, when I joined Dr. Aaron Mitchell's lab a while ago now, as a postdoctoral student, I was thinking about biofilms. What can I study in biofilms? How can I understand how biofilms form better? Now, I would be a liar if I didn't say I also like to tinker and I love my gadgets. And so I was looking around the lab, and then in the corner there was this BioFlux 200 machine. And I got very interested because with this machine you have a heated stage, so you can control the temperature at which your experiment is going to happen. You can actually cause constant flow rate, and it's very well regulated so that you know that it's reproducible. So every time I'm doing this experiment, I know that that rate is always going to stay at the same flow. And most importantly, you can actually visualize what's happening. So if we take a look here, basically here's kind of one well of the BioFlux system. You can basically insert your sample, right? And then as you apply pressure, it moves down, it moves across, and exits. Now on the bottom here, we have a cover slip, which allows you to use a microscope to actually visualize this. So you can actually see as these cells are being pushed through here on flow. Now another actually added benefit was actually this material right here is very similar to catheter material, which is one of the primary sources in which we find biofilms forming. Okay. So with the ability to visualize this, I thought that we could use this machine to assay how biofilms form. So I started out with a bunch of different techniques. And again, one of the great things about this machine is you can actually take video. You can take pictures, you can take video, and it can allow you to see a bunch of different aspects of biofilm formation. So I started out with, well, let me put my cells in. I would first put the cells in as yeast. I would add media, spider media, which is inducive for forming hyphal cells and forming biofilms in vitro. And I had it set to 37 degrees, which would be your ideal biofilm formation conditions. And as you can see here, you can actually observe the the hyphal cells growing at quite quick rate. They're dividing, they're diversifying. Now, unfortunately, as we move along here, let me see here, wait a second, you can start to see that you get some cells starting to clump up on different segments of the biofilm. I'm sorry, of the cells here. So what we're seeing right now is basically the yeast cells being pushed from upstream and binding to our very sticky hyphal cells. So while this provides a nice little video, it was hard for me to envision how this could be quantitative at the time. Well, that being said, you definitely could grow a rudimentary biofilm. One of the limiting factors is the depth of the well. So this actually prevents you from getting up to that very mature biofilm. But that being said, you could still see the rudimentary beginnings of how biofilm forms. It also allowed you to observe yeast and hyphal cell adherence, how they stick to the surface, and how they then grow and divide from there on. One of the difficulties there, that last video you were looking at was about 18 hours, was keeping the focus. Basically what you saw was me coming back about every hour, 6 hours at night there, to refocus so that everything would stay in focus. And the other problem is as you have hyphal growth, you have hyphal growth growing up, growing to the sides, basically every single direction. So you can't have everything in focus. Now, that being said, they now do have a fixed system. They fixed this problem with an automated system that's now available. And I will say that Dr. Clarissa Noble at the University of California, Mercer, I hope I'm saying that right, has used this successfully for studying biofilms. And in fact, if this is of interest of how this can be used to study fungal biofilms, I highly recommend you take a look at some of her papers, as she's done some great work on this. So, but for me at the time, I was sort of thinking, well, if I can't use this to really grow a full, mature biofilm, and even if I could, it's just sort of a video, what else can I use this for? What else? How can I use this to be a quantitative assay to better understand how biofilms form? Well, at the same time, Dr. Mitchell and myself were in the process of writing a book chapter and a review on biofilm formation. And one of the things that we did is we kind of took this in vitro model of ours and started mapping out where does everybody map to? Where do the transcription factors, the different genes, what do we know about biofilm formation and at which step? And one of the things that jumped out at me right away was, well, we knew very little at that point about the very first step of biofilm formation, adherence. And in fact, we had one gene or one protein that functioned by inhibiting adherence. So it wasn't even the step that would allow you to start the whole process of biofilm formation. The other reason that this first step of adherence interested us is that, again, as you move from left to right here, you're having increased drug resistance. And if we can understand the mechanisms by which the fungi can first start out this whole process, in theory, we could be able to identify markers that could result in better drug discovery and better treatment options. So, we wanted to identify genes that govern the adherence step. Well, there's a couple different ways that I went about this. So, first off, I said, okay, well, I'm going to look at conditions that are ideal for growing yeast cells, and that would be 30 degrees Celsius, and again, at a certain flow rate, at a fixed flow rate, sorry. And then sort of the thing was, well, what can we see? So, some of the first stuff was kind of looking at how do we see adherence. So, very happily, we see adherence, but one of the limitations we quickly ran into is that the amount of cells that you add has to be carefully regulated. Basically, because if you add too much cells, as you can see what's happening here, you end up with clumping, and it's very hard to quantitate how many cells, for instance, are in that clump that I'm pointing to right now. But now, if you take it a little better and you modify this and you use a lower amount of cells, you can see, such as we see here. And here we have yeast cells sticking as they come along, and you can actually see, okay, well, there's 1, there's 2, 3, 4, 5, 6, and so on. And so we have sort of a platform now where we can look at just single yeast cells adhering to this catheter-like substance within the machine, or within the device, I should say. Now, excited about being able to do yeast cells, I said, well, what about hyphal cells? Let's take a look at hyphal cells. Well, hyphal cells weren't quite as easy and actually are a little more difficult to do here. And that comes into a general problem where, as you can see, we have these large clumps of cells. Hyphal cells are very, very sticky. So first off, basically, anyone that's there wants to stick to them. It's hard to develop a condition where every single cell is a hyphal cell versus a mix of hyphal and yeast cells. And because of that, you have these huge clumps that can occur. Another problem we ran into is that the hyphal cells would end up actually starting to block the channel, so giving us basically, making it hard to run the assay. So with that determined, I started to perfect an assay to look at just yeast cells. And what I found worked best was to use 500 microliters in the sample of YPD. Now, this is a yeast that was grown overnight and then diluted to an OD of 0.2. And basically, we set at a flow rate of 3 dynes per centimeter squared for 30 minutes. Now, the reason we did this flow rate is that I discussed with some medical colleagues and some literature, and this seemed to be sort of a good flow rate for what could be considered a blood flow rate, the flow rate we would see within blood vessels. So basically, I would add my cells, I would turn on the flow, And for 30 minutes, yeast cells would fly through, and some would stick and some would pass through. And at the end of 30 minutes, I would take this and I would take pictures along the lane of the well. And obviously we do this in duplications. Now I found it was very important for reproducibility to have multiple pictures per well or per channel here. So this would be, for instance, I would count this as one. I'd have two pictures. In this case, I would count up all the yeast cells and I would count that as the one for the one sample, if that makes sense. So I would count, 1, 2, 3, 4, 5, 6, 7, 8, and that. So multiple pictures within one well, total amount of yeast, and that would give me a number for which to base things off of. And to give you more of a visual representation of this, for instance, this would be one picture, one of 2 or 3 that I would take. I would count up all these different yeast cells, and that would be the number for one well, right? Do it 3 times, divided by 3, and that would give me my sort of wild-type number of cells. Now, if I were to look at a mutant, I would see, obviously, in this case, much fewer cells. And this would indicate that the mutation, or the gene in which this mutation is responsible for adherence of yeast-formed cells. Now, what was very nice about this is it was highly reproducible, and gave us some very great information. Now, I want to stress that for people who are interested in doing this, it's important that you use a wild-type strain for every new plate that you use. That gave us the most consistency and most reproducibility. So we started out by looking at 197 transcription factor mutants that we had in the laboratory of Dr. Mitchell. And what we found was actually a little surprising. We actually found 30 transcription factors that regulate adherence. So what we're looking at here is the relative adherence with our wild type being basically our 100% or 1, and then our 30 transcription factors who affected adherence. So for instance, as you can see here, ZCF31 had around 40% of the amount of yeast adhering compared to the wild type. And all of these were statistically significant. Now, we were pleasantly surprised, I'll say, by the number, but I think this also stresses the importance of adherence for the life cycle of Candida albicans. Now, to summarize this, basically this assay is highly reproducible, and it's a high throughput assay. So we were able to look at 197 different strains, or mutants, 3 different strains for each. And we're able to do this in a relatively short time once the kinks have been worked out. We're able to identify 30 transcription factors that are required for adherence. Whoops. And from this work, we've actually been able to start to shuffle out and learn new pathways for what's occurring with biofilm formation. So for instance, BCR1 is a transcription factor that positively regulates ALS1 that results in adherence of yeast-formed cells. SNF5 is a transcription factor that regulates the transcription factor of ACE2, which regulates SUN41 and PGA10 to allow for adherence and also play an important part in initiation. Now, this was where my postdoctoral work ended. One of the things that we were able to do with, actually before I get to that, let me just say that with the transcription factors identified, we then did genome expression analysis for each of the mutants that had reduced adherence, and that's how we were able to discern some of these pathways. It was great work that was done by other fellow postdoc in the lab, Wenjie Zhu, and we were able to learn quite a bit from it. Now, at this point, an interesting opportunity was presented to me. How would I like to go to the Middle East, Qatar, and help form a biological science department and do research and get to teach courses as a professor. And this was an excellent opportunity. I was newly married, and I took it, and I'm very glad that I did. Now, when I got to Qatar, one of the things I wanted to stress was that I was going to study things that were not important only to the United States, but were also important to Qatar. Now, fungal infections in the Middle East are actually of great concern. And now, this is due to the increased use of implanted devices and the rapidly growing population of diabetics. We'll say that, many of the Middle Eastern countries, particularly Gulf countries, have in the last 50 years resulted in an immense amount of wealth, but they've also used that money to pay for and treat implanted devices that previously they were unable to do. And in fact, their rate of treatment and medical treatment, I should say, is quite high given the amount of money they have to basically treat patients. Now, Candida albicans is still the primary source in Qatar for this, but there were other less studied fungal species that were being isolated at increasingly at a rapid, they were identifying other fungal species, and the rate at which these were being identified were increasing at a very rapid and concerning rate. So I went down to the major hospital, and I asked them, what is the important things? And he said that while Canada was able to meet with a fantastic researcher, mycologist, I should say, at Hamed Medical Hospital, and there he said, Dr. Saad Taj al-Din, and there he pointed out that these other ones are more concerning. For instance, the trichosporin species infections had increased in Qatar tenfold in under 7 years. Now, this was a little interesting because trichosporin species, in theory, shouldn't have been in Qatar. And one of the hypotheses that I have for the increasing rates in these rare, quote-unquote, rare fungal infections, or fungal species infections, was that there was increased treatment with the catechindins. Now, catechindins aren't expensive, but they're very effective against treating Candida albicans in particular. Qatar has a lot of money, and they're able to use catechindins versus the more, let's say, inexpensive azoles or amphotericin B to treat these infections. So what was happening is basically you were eradicating Candida albicans, but again, potentially leaving a vacuum for these other fungal species. So my question was, obviously as a person now that has become very interested in adherence, was how does adherence differ among the different fungi? Now, one of the benefits of having a mycologist that's working at the largest hospital in Doha is that I had access to patient strains. And so the question was, what are we seeing within a certain patient of a certain fungi, and how is its adherence differing? Now, luckily, Dr. Saad Taj al-Din had saved strains, I guess you'd say, for the last 10 years of patient. They're isolated from Hamed Medical Hospital. Now, using proper protocols, I was given strains with no information of patient, all that good stuff. Now, he was also very good about recording the location of infection. And he had actually started using, at that time, a fairly new technique using multi-top mass spectrometry to identify the exact species of fungi. Now, this is important because fungi can be notoriously difficult to identify the specific fungi, and often cases in a hospital clinical setting, it's more important to know the genus of it, Candida species, because the treatment is very similar from one Candida species to another. Whereas by using the multi-top mass spectrometry, you have a very high rate of identification. Now, we started out by thinking, well, let's assay the strength of adherence compared to our known reference or type strains. Now, while we started to do this, we were lucky enough that there was another researcher studying the appearance or the development of fungal infections in patients with cystic fibrosis. Now, this is the most common genetic lethal disease that affects Caucasian populations. So I was a little surprised when I heard that they were studying this. But basically because it's observed in low frequency in the Middle East. But there are a number of families in Qatar which actually ends up having with this with high frequency. again, with the good medical care available, there were constant swabs of the mucus and culturing of this to know what bacteria and fungi were occurring in there. And again, we knew the specific species because of the use of the MALDI-TOF. Now, just to help remind everybody, cystic fibrosis is a defect in ion transport across the membranes, and it results in mucus that is thicker than usual, and it builds up in the airways. This mucus is an excellent place for bacteria and fungi to grow. And repeated infection actually results in damaged lungs, which also provides a route for both fungi and bacteria to become more systematic within basically worming their way through this damaged tissue. Patients often die of infection or respiratory failure. Now, the real interesting thing was that in Qatar, the most frequently occurring infection was with Candida dubliniensis. Now, this was surprising because, every literature that we looked at would say Candida albicans, Candida albicans, Candida albicans. Well, Candida dubliniensis is very similar to Candida albicans and was in fact only identified as a different species back in 1995. They have very similar morphology and a similar genomic structure. So the question that had to happen to us is, if everyone's telling us that Candida albicans is the primary, why in Qatar are we seeing Candida dubliniensis? And of course, being the person who loves adherence, I said, let's take a look at adherence. Here's one thing we can look at. So when we look at the Candida, so here we have, again, a relative adherence. Here's our type strain for Candida albicans. And then we had 5 patient strains. So what's first thing that kind of jumped out at us right away was that patient strains do not all have the same level of adherence. We were very surprised when we looked at the Candida dubliniensis. Again, here is our type strain, which has almost double the amount of adherence of Candida albicans. Again, we see a variation occurring here in the adherence from patient strains, but we see for the most part the majority of the strains being statistically significant higher adherence than that which we saw with the patient strains of Candida albicans. Now, we also had some tropicalis, Candida tropicalis, and again, we didn't see any increased adherence, but we did see, again, the sort of varying levels of adherence within the species. Now, this was quite fascinating. We were able to publish this, and the data suggested that the presence of Candida dubliniensis in the lower airways of Candida, what happened here? Okay, I think I'm back now. Okay, sorry. The data suggested the presence of Candida dubliniensis in the lower airways of cystic fibrosis patients may be related to an increased adherence properties. Now, many clinical isolates, one of the other things we knew, was that many clinical isolates are misidentified as Candida albicans when in fact they are Candida dubliniensis. And one of the reasons for this could be that Candida dubliniensis is hard to discern from Candida albicans, and treatment is the same. So if you have a Candida albicans versus a Candida dubliniensis, who cares? We're still going to treat it with an echinocinin or at least an azole whatsoever. So one of the hypotheses we have is that it's possible that Candida dubliniensis is in fact more prevalent in cystic fibrosis patients. It's just been misidentified. Now, the other interesting thing is that we found Candida dubliniensis would co-inhabitate cystic fibrosis lungs with bacteria that were known to be detrimental to Candida albicans. So in many of the more adherent Candida dubliniensis patient strains, they were also isolated with either Staph aureus or Pseudomonas. So this indicates that they may have a way to overcome the mechanisms that Pseudomonas and Staph use to basically attack or prevent Candida albicans from establishing an infection. Now, this work kind of pushed us a little further to think about other rare fungal infections. And again, historically, Candida albicans has been isolated in 70 to 80% of all infections. But interestingly, this number has begun to decline. So in some cases, we see numbers now getting closer and closer to 60 or less % of all infections being due to Candida albicans. Part of the reason for that is more accurate identification of species, but is also the resistance of other species to specific antifungals, both innate resistance and then acquired resistance. The other reason that we're suddenly seeing rare fungal infections is that we've seen an increased rise of global travel that exposed people to tropical or unique environments that they usually would not have been. Myself being the case of traveling to Qatar. I didn't get an infection, but as an example. Now, so what we decided to do is let's take a look at a bunch of different pathogenic fungi and see what their adherence is by looking at their type strains. And as you see here, again, we have Candida albicans as sort of our control or our reference. It comes at 100% of adherence. We compare everybody from him. Candida glabrata, very similar to Candida albicans. Tropicalis had a slightly lower. But as we move along here, we saw that a number of the trichosporon species, very interestingly, had great reduced adherence compared to Candida albicans. So I said, okay, well, fungal type strains, can we use this as a way to identify potentially that the fungal species or genus based off of adherence? So, trichosporon. Well, trichosporon species were of interest to us because they're the second most common non-Candida fungi that cause invasive infections. And there was a huge rise of them in Qatar. Now they were, if you look at the textbook, they're mostly isolated tropical regions, and they are associated with superficial, subcutaneous, or systematic infections. Now, tropical regions. Now, the funny thing is, after I started working on this and talking with some fellow colleagues, you started to hear like, oh, no, we've seen trichosporon species, including my old alma mater in Madison, Wisconsin. One of my colleagues said, oh, yeah, no, I've seen 2 trichosporon species infections here turn up. So I think the original belief of, this is only in the tropical species, that, this species is only in Europe, I think international travel has kind of negated that belief, or negated that sort of definition, but would also indicate that these species can survive in alternative environments than what was previously thought. Now, treatment of trichosporon with azoles can be effective, but it requires the recovery of the immune system. And since the immune system may take a long time to recover, you run the risk of resistance. Trichosporon species have innate resistance to all forms of echinocandins currently. So using the type strains again, we looked at a number of different species. We looked at Trichosporon dohanus, which was actually isolated in Doha. It's its own unique species. Trichosporon asahi, which is the most common trichosporon species. You have Mucoides, Gepiconum, and so on. And we found that, in fact, they all had reduced adherence compared to Candida albicans. Now, type strains are great. They're perfect because they're a type strain. We can use them, and we have a general idea that this is our wild type strain. But the question came in is, what about when we look at patient samples? So Dr. Saad Taj al-Din, again, had done a great job of cataloging and maintaining all these strains. And what he was able to give me was quite a few different strains, including 21 Trichosporon asahi strains. And when I assayed these for adherence, I was shocked to see how much difference we see in the level of adherence from one patient strain to the other. And that doesn't just stick with Trichosporon asahi. We also see that with Incans and with some of our other species also, including Bacillus and Gepicombs. Now, what does this all mean? Well, the undergraduate who was working with me was in tears. She thought that she had found nothing, and then now she had wasted a year of her life, etc. But I said, let's dig into this a little bit more. What can we find? Now, location was something that Dr. Taj al-Din was very good about keeping record of. So when we started to take these species and we ignored the species and instead focused on the location, that's when we found some very interesting preliminary data here. And what we find is that in urine, samples collected from urine, sorry about that noise, background noise, samples collected in urine, we find lower adherence rates compared to the reference strain. Whereas when we look at the nails, those isolated from nail infections, we see increased adherence, as we see with ETT, which is our endotracheal tube location. So they swab the endotracheal area, and from that you can see. And, these are quite increased quite a great increase in adherence compared to our reference, our Candida albicans strains. So in concluding with these results, which we're in the process of publishing, we were able to see that variable changes can be seen within species and from species to species as far as adherence is concerned. But that we it appears that we have location has the consistency, so that the location in which a fungi is being isolated will have a consistent level of adherence. So for instance, urine results in decreased adherence. One hypothesis we have about this is that, in fact, what we're seeing is that we're selecting for those cells that are less adherent. So if you're very adherent, you're going to stay stuck to either the bladder or the kidney as you're being pushed out at high flow. If you're less adherent, you're the ones that are going to most likely end up coming out. Now, ETT and our nail, endotracheal tube, and the nail have increased adherence. If we think about this, nails are keratinocytes, they're dead cells. So there is, again, no immune response when they get bound to. And the same thing happens to your endotracheal tube. The tube is itself a plastic or silicone-based type device, which, as we know, is a perfect reservoir for forming a biofilm. Now, the stronger you can stick to those devices, the more likely you are able to form a colony or a biofilm that can result in prolonged infection occurring. Now, that ends my presentation today. There's a couple people that I really would like to thank. You can't do work like this by yourself. I'd like to start by thanking Dr. Aaron Mitchell at Carnegie Mellon University. He was a fantastic postdoctoral mentor, and I can easily say that I would not be where I am studying what I am studying if it had not been for his excellent mentorship and continued guidance. Just to give a little plug, I don't even know if he's looking for him, but if you are looking for an excellent mentor as a graduate student or as a postdoc, you could do no better, than Aaron. Dr. Carol Wolford, Dr. Wenji Yu were both, Wenji was a postdoctoral fellow with me, helped with the expression analysis. Again, couldn't have learned it like what we learned without him. Dr. Wolford is a staff scientist who helped greatly with creating the mutants. She's got the touch for that. And David was an undergraduate, and I guess I have to call him Dr. Wang now. He is starting his residency in San Francisco. Now, while in Qatar, Dr. Saad Taj al-Din was able to help me out with the collection of the cells, interpreting of data, and I had a bunch of great undergraduate students I want to mention, including May El-Jindi, who is currently a graduate student at Yale. I'm very proud of the great work she did. She was the one that did the primary amount of work that you saw with the adherence. In addition, I had some great students, Raji Khatibi, Ridden, Rula, and Fatima. I'd like to thank the University of Detroit Mercy for providing me research space and funding to continue my work, Carnegie Mellon University and Carnegie Mellon University Qatar for, again, funding research. NIH helped fund this work through the National Research Service Award and an individual postdoctoral fellowship. And I'd like to thank Qatar Foundation for the award of an NPRP, which is the equivalent of an R01 in the United States. And at this point, we'll take any questions. Thank you for listening. Hopefully this was helpful.
This is a remarkable talk. Thank you, John. I want to start out with a question related to adhesion versus virulence, or adhesion as it relates to virulence. Where do you see that trend going over the next 5 years?
Well, for the next 5 years, it's sort of been doing it for a couple million years, I'd say. But I would say with adherence, sorry about this background noise. I don't know what's going on. Okay, there we go. I would say adherence is essential for virulence. So when we look, there's been a lot of great work done in, for instance, the laboratory of Dr. Scott Filler in California, where he has shown that the yeast cells will enter the bloodstream, and as a low nutrient condition, this actually causes them to stick filament, and then basically those filament either will go into the cells or between the cells to basically enter a more rich environment. So adherence is definitely tied to virulence. If you knock out adherence, you do see, for some of these strains, we be able to look at, we do see a reduction in virulence. And you definitely see a number of these strains unable to form biofilms. So without the ability to form the biofilm, you kind of can remove that reservoir where continuous or systematic long-term infection can occur. Is that what you were looking for? Sorry.
Yeah, that's what I meant in terms of the trend, meaning the research trend of where we'll see where virulence and adhesion sit. One of the interesting trends. One other thing to add,
go ahead. Sorry, one thing to add there. Sorry to interrupt. Is that I think the further study of how do the cells, when they come in contact with human cells or with their host cells, what does that trigger as far as adherence? What are the conditions that can say, okay, now I need to adhere, now I shouldn't adhere, are things that are of great importance and that we'll be learning more and more of as we move along.
One of the interesting trends that's come out of biofilm research recently has been this concept of cooperation versus competition. And, when I went to school a while ago, we were taught that the way biofilms were formed is that the bacteria basically hold hands and they walk into the woods singing songs, and they make biofilm. And now the trend seems to be pushing more towards the concept of competition. And Kevin Foster's group at Oxford published a really interesting paper about this, suggesting, one of the reasons that Pseudomonas is able to form such a strong biofilm is because of the endogenous antibiotics that are produced. Do you see that being something similar in terms of in the fungal side? And I guess the second part to that question, and this is actually even more interesting towards the bacterial side, is that antibiotics can trigger biofilm infections and actually make them worse before they eradicate everything. Would that be the case as well with fungal?
Well, for the first question, I would say, when you take patients, when you collect, let's say, from bloodstream biofilms and stuff like that, the cells are mostly clonal. So you're all coming from basically the same genetic background with the fungi. So they are definitely working together. There's definitely not it's not, I'm going to do it all on my own. It's a clump of cells that come together and function and trigger basically, we have to form a biofilm here, or this is a good spot to form a biofilm, in a sense. Now, they have tons of competition. The microbes do not live in a vacuum. They live among each other, and they compete with each other. Sometimes they work together, and sometimes they are detrimental to each other. Pseudomonas is definitely known to attack Candida albicans in the filamentous hyphal form. And some of the other bacteria have been shown to release quorum-sensing molecules that prevent them from forming filaments, which would then, basically prevent your ability to form a biofilm. So thus reducing competition for resources at that particular spot. As for, what was the second part of your question again? Sorry.
Well, it was a continuation of that where one of these mechanisms for competition as a driver of biofilm formation has been endogenous antibiotics. And I've seen myself, at least with Staph aureus, for example, that if you give a sub-MIC of Cipro, that you basically explode the infection. Now, of course, antifungal is working completely different mechanisms than antibiotics do, or remarkably different, I should say, not completely. Do you see that being a case with fungal stuff?
Well, I will say that one of the things that we do know about fungi, Candida albicans in particular, is that when stressed, one of the things that can happen is filamentation. So if you're not giving enough of a drug, you are stressing that community. And so I would think that would definitely, if you're giving the wrong MIC for a particular infection, that would definitely cause them to become, I don't want to say it would cause the infection to explode, but could cause them to become more, push them more towards virulence from the commensal form, if that makes sense. I would say also one of the bigger concerns with the azoles, for instance, is that if you're not giving the right concentration, you're stressing the cells, and the prolonged exposure to the azoles is what leads to the azole resistance that is so problematic with the patients that have long-term infections that need to be constantly treated.
And a more technical question it relates to being able to, how do I say it? With the BioFlux system, to be able to capture that moment from where you've got a budding yeast cell on a plate to driving hyphae growth and then potentially a biofilm formation. Do you have any suggestions on ways to do that help clean up the assay a little bit?
Yeah, I think one of the things that could be kind of interesting using the assay, going forward would be, in theory, you should be able to create a very single level, gross the HeLa cells or some other sort of human cell line, and then basically throw the fungi at it and see what happens and look at different mutants. I would say the other potentially interesting thing is that you have the Fluxion, which has the two different wells from which you can pull reservoir for you basically change the media. And, obviously the big interesting thing there to me would be is what happens if you change from azole, a media with azole, so you establish this kind of rudimentary biofilm, you treat it with azoles, and then you pull back and you treat again, what sort of effects you can have. Basically, you can use any sort of compound to test, can I knock these yeasts off of the silicone background or the thing that they're adhering to in the machine by basically adding it after they've adhered. Does that make sense?
Yeah, it does. And I want to thank you again for this great talk and congratulate you on this very impressive body of work. I think that concludes our webinar. Anything else you want to add, Dr. Finkel?
No. I hope it was informative. If anyone has any questions, please feel free to email me at the University of Detroit Mercy. It's finkeljs@udmercy.edu. And I'm happy to answer any questions I can. And I want to thank Fluxion for giving me this opportunity. And Ryan, thank you